with our next session. Welcome, everyone. I'm Joshua Schimmer from Piper Jaffray, one of the senior research analysts at that group. Spent a lot of our time in gene therapy and cell-based therapy, and we're going to talk about some of the programs and opportunities that await the platform in oncology. So maybe we do have a diverse repertoire of companies represented, so maybe the best strategy to start with is to have everyone kind of introduce themselves, their area of focus, their cellular therapy approach, and then we'll try to tie together a Q&A that integrates everyone's experiences. So Oz, why don't we start with you?
Hi, good morning, everybody. My name's Usman. Everyone calls me Oz. I'm the head of the cell and gene therapies business at Novartis.
Hi, Jeff Abbey. I'm the president and CEO of Argos Therapeutics, and we're in a pivotal phase 3 trial for metastatic renal cell carcinoma with a autologous cell therapy.
Well, good morning, everyone. My name is André Choulika. I'm the chairman and CEO of Cellectis, and we're a CAR-T company focusing in immuno-oncology.
My name is Gregg Sando. Good morning. I'm the founder and CEO of Medica. We are developing autologous T-cell immunotherapies for both cancer and infections. Our lead cancer program is aimed at oncogenic viruses, and we're in phase 2 in the US, Europe, and actually South Korea in an international trial. Thank you.
Well, I think the devil is always in the details for a lot of these programs. So Gregg, why don't we stick with you and maybe elaborate a little bit on your strategy, the type of cells that you work with, the modification approaches that you use, and then ultimately the context in which they get delivered.
Okay, so our strategy in oncology is really to target cancers associated with oncogenic viruses. And our lead program is aimed at the Epstein-Barr virus, which is actually associated with a range of different malignancies, including lymphoma, nasopharyngeal carcinoma, and gastric cancer as well. And we looked at a number of different cell therapies probably about 6 years ago now. We've been operating as a T-cell immunotherapy company since 2007. And we were particularly attracted to this space and this technology, which was under development by Baylor College of Medicine, because we felt that targeting oncogenic viruses gave you the optimum balance of safety and efficacy. The cells that we target with our activated T cells, the cancer, is expressing viral antigens. These are non-self antigens, so that we don't have on-target, off-tumor toxicity in our applications. Our cell therapies are very safe, and this particular approach is very safe, and we've shown that, and Baylor College of Medicine in their research has shown that through treating hundreds of patients. So we can get a high affinity targeting effect by using T cells which recognize viral antigens on cancer cells, but we can maintain the safety of that treatment as well.
Great. André, maybe elaborate a little bit on your use of gene editing and incorporation into cancer immunotherapeutics.
Yeah, so we use like T cells, like the propency of T cells in killing other cells, especially like infectious cells, in order to turn the T cells into a cancer-killing machine. And also there is a second thing in T cells is that they amplify. These are the two parameters that make CAR-T therapy extremely powerful. So with a CAR, the advantage of a CAR is that you can kill a cell through the CAR with the antigen very lowly expressed on the surface of the cell. For example, a monoclonal antibody naked can kill a cell if the cell expresses the antigen over 100,000 molecules on the surface. For an ADC, it's approximately over 10,000 molecules on the surface. For CAR-T, it's probably maybe a handful. So it's very powerful technology, and also like the risk behind this is to target also healthy tissues, and everything inside a company is trying to gene edit the T cells and removing or replacing certain parts in the T cells, first to make them off the shelf, and also the cells compatible with other type of drugs, because we're definitely convinced inside Cellectis is that combo therapies, it means potentially combining like chemotherapy with CAR T cells, is one of the keys into treating certain forms of leukemia and also a pinch into like solid tumors in general. So gene editing is one of the big parts of cell therapy and specifically CAR T therapies.
Great. Jeff?
Sure. So we are targeting the patient's own tumor-specific neoantigens by capturing RNA from the patient's tumor. And we're taking all of the messenger RNA, so all the encoding RNA from the patient's tumor, in order to capture all of those unique neoantigens specific to the patient's own tumor, and programming a highly optimized dendritic cell, which we make from the patient's monocytes. So it's 2 autologous collections to get this highly optimized dendritic cell that's expressing all of those tumor-specific antigens. And what we see in the patients are a dramatic increase in tumor-specific T cells, which, similar to what Gregg described, are only targeting the patient's tumor. So we see no collateral damage at all. And this obviously has good safety toxicity profile and allows us to combine with other effective therapies that have different modes of action. So in our phase 3 trial, we're in metastatic kidney cancer combining with standard of care, and we're just going right on top of standard of care. Obviously, in earlier stage diseases, we can go as a single agent, but in other disease settings, we can combine. And this approach can be utilized, in virtually any tumor type, because all we need is a tiny amount of cancer cells to capture the RNA and then program the dendritic cells.
And as you probably have, maybe because of the deep pockets of Novartis, a broad perspective, not just to focus on what you guys have going on, but to also kind of scan the universe for opportunities that you might like to add to the armamentarium. So, I guess I'll let you answer kind of the question how you want, whether you want to tell us what you have going on at Novartis, or whether you want to tell us what it is that you look for in emerging technologies, or maybe a little bit of both.
Sure, happy to do that. Good morning, everybody. So from a Novartis perspective, clearly the title of the panel being focused on oncology, clearly we are heavily invested in immuno-oncology, broadly from a small molecule perspective, biologics perspective, but also from a CAR T perspective, which takes up probably 80% of the time of my organization on trying to make CAR T therapies a reality and bring those to market as soon as possible for patients who frankly have no other alternatives. Aside of that, within our organization, we have a very active interest in graft versus host disease. We have an excellent collaboration with a company called Regenerative down in the University of Kentucky in Louisville, a very exciting program to really look at how you can actually eliminate immunosuppressions in the setting of solid organ transplant. Very exciting area to work in. We also have a very active interest in programs in human stem cells. And from a research pipeline perspective, with my colleagues in research, obviously immuno-oncology targets are a big focus. You may have seen some news flow around that this morning as well, with companies that we're collaborating with. In addition, we have programs heavily vesting in ophthalmology, back of the eye, retinitis pigmentosa gene therapies that we're very interested in. We're also looking at sensory neural hearing loss, with regenerative therapies there as well in the form of AVV vector technology. So from a broad platform perspective, Novartis is scientifically agnostic when it comes to the entire field. However, we do feel that hematological malignancies has made the most scientific and business focus in the past few years. And that builds on the previous panel that I think we've come to a maturing and thinking now that certainly in the area of CAR T therapies, there is overwhelming benefit risk and now capabilities around manufacturing science and scalability that will allow at least a very strong beachhead to be formed for this industry with regards to CAR T therapies and hematological malignancies. Our vision at Novartis is clearly that will allow further scientific investments, not just by Novartis, by other big players, and I would encourage those and other larger organizations to have the courage that Novartis did in terms of dedicating resource and creating specific business units around this. So it's a thrilling time to be in this space. It's an area that is a big focus for Novartis, and we hope to have some good news flow in the 2016-2017 time period with the advances of hopefully the first approvals with CAR-T therapies.
Great. Maybe for Gregg and Jeff, I guess from my own perspective as a research analyst, we spent a lot of time historically on small molecules. Then it got a little bit more complicated, and it got to bispecific antibodies, and then cells and viruses. It's getting kind of technical, and it feels like there should be really high barriers to entry, and yet there are a lot of companies moving into the space. So maybe from your experience, can you talk about some of the technological scale-up challenges that you faced in the small companies? What was it that enabled your groups to accomplish that? I guess I'm trying to reconcile the complexity, which would seem like this is territory that only deep pockets like Novartis can really get into, and yet we have a number of small emerging companies that are doing very exciting things. So Gregg, why don't we start with you on that one, and Jeff, you can throw in.
So I think there are two key questions that all the companies up here and working in this space face. The first is, do these cell therapies work? And the second is, if you can answer that question in the positive, can you actually scale up and manufacture these cell therapies to treat thousands of patients? So the first question is obviously under intense study right now, and a number of clinical trials, a number of companies have taken on this challenge. But in the big picture, what we're doing here is essentially just using the human immune system as it has evolved to behave, which is to control infections and cancer in the body. I think we all realize at this point in time that our immune systems are keeping us free from cancer on a daily basis, and really when a malignancy develops, it's because it's found some way to evade the natural immune response. But there's a lot of work we need to do in order to really understand how to harness the immune system and make it work on demand, so to speak, in these cell therapies. The second challenge, can we manufacture, is a big one. And I think in our case, we're just very lucky that we started working on this in 2007 because it's a big challenge. And I think we were lucky to get a grant from the UK government. We're based in London. That helped us to build a closed system manufacturing approach for our lead cell therapy. So it takes a lot of work. There's a lot of problem solving for manufacturing. It takes time, and each manufacturing system has to be developed specific for the cell therapies that you're working with. But ultimately, we strongly believe we'll solve the manufacturing, and indeed in our lead product have solved the manufacturing in terms of a scale-up approach. But there's a lot of work and a lot of innovation that still needs to be done here.
Can you maybe elaborate on that aspect of development? And when you say there is a lot of work, what's involved with that scale-up?
Well, typically these cell therapies start in an academic environment where you're operating under a biological safety cabinet, where your hands are sort of stuck underneath the cabinet, and you've got the clean air environment behind you. And that's basically like trying to manufacture something with handcuffs on. It's pretty close. So what you need to do is actually take your manufacturing processes and put them into a closed system. At least this is our approach, a bag system where you can manipulate the cells in a much more free environment and with air systems that are much less costly, and allowing your technicians to multitask and work on multiple products at one time and take them through essentially an assembly line. This is all possible, and then once you build that assembly line, you can introduce increasing automation to it, just as people did with manufacturing cars a long time ago, the old-fashioned cars, that is. There's I think, a clear path forward, but there's just a lot of work that needs to be done at this point.
Great. And Jeff, maybe talk about your own experiences, and we could also kind of throw in the IP landscape and whether that's been difficult to negotiate.
Yeah, so we started in 1999 and didn't get into the clinic for the first time until 2004 because we looked ahead to that. I agree with Gregg, right? There's the two issues, does it work, and then can you manufacture? We decided we wanted to make sure we could manufacture at the same time we were testing to see if it worked. So what we took from our academic collaborators back in 1999 was a process that was being used at their universities, where they would take fresh cells, take fresh tumor, make the product, and immediately put it back into the patient. And we said, well, that's not going to work commercially, let alone in a phase 3 global study like we're doing now. So we needed to develop a process where you could take cells that were at least 18 hours old and be able to store the tumor that you were getting, which has to be fresh, but develop a process to isolate RNA from frozen tumor. And it wasn't until 2004 that we were ready to go into the clinic in our first clinical study. And then we learned a lot from that study. We actually improved the process to make it more potent from that, and then went back into the clinic again after that. But I think Gregg hits on that key point, which is you need to think about the manufacturing way ahead of time in terms of, can you deliver this to patients all over the world in an effective, cost-effective, and viable manner. And so what we wanted to focus on was being able to take that one cell collection and make multiple years of doses, so that hopefully if it is effective and patients are living long, you only have to collect their cells and do this once and treat them, for a multiple number of years. And in our phase 3 trial, it's worked out beautifully, where we're averaging 3+ years of therapy from one collection, and able to do this globally at 130 clinical sites, all shipping into North Carolina and shipping out of North Carolina. But it took years and years of development to get there. So I think the barrier of entry, to answer your question, is not just on IP. There's so much in the way of trade secrets and other knowledge that these companies like ours have developed, which isn't necessarily captured by IP. But in our case, it turned out that we now have a patent that protects using monocytes that are at least 18 hours old to make a antigen-presenting cell. So that IP came out of development that we knew we had to do to be able to make a procedure that would work on a centralized manufacturing basis, and ended up developing IP and getting it patented, which protects the business model, if you will, of using cells that are at least 18 hours old.
Great. André, maybe you can discuss the experiences with an allo approach and scaling that up, and what challenges might be unique to those efforts, because I know you've been very active over the last year.
Yeah, I'm glad you're making this focus on manufacturing, because I think in cell therapies in general, it's one of the main hurdles. And of course, you have two options with cells. Either you want to reconstitute an organ, then it's better to go for auto. And in the case of immunotherapy, where you have to destroy a tissue, it's better to stay allo and get rid of the product at the end of the operation itself. And for the case of CAR T therapy, we took this allogenic approach, where you can manufacture frozen pharmaceutical product that you can store, ship all over the world, and can QC, QA, can monitor, and all these things. So now we're starting from cells, the raw material, it's primary T cells. You don't have a cell line that is a T cell, actually, because these T cells have to amplify and kill, and a T cell can kill in a row approximately 5,000 to 10,000 tumor cells, and can amplify by 10 to 100,000 fold. And once they've achieved this amplification and killing, then they're exhausted and die. They cannot remain in culture forever. So this is the main problem. So what we do, we start from primary blood cells, so apheresis. So we have T cells inside, so we activate the T cells, and then you need first to add genes inside, which is the CAR, maybe a suicide gene that we do. And then the step after, we have to remove certain genes inside a T cell using gene editing technologies at a very precise and without being able to alter the quality of the cells in having too much off-target cleavage inside a T cell. And we use a technology called TALEN, which we think is extremely safe for the T cells and very powerful. It means like the number of modified T cells is pretty high. The goal at the end is to have like a cost-effective product at the end. So step one, you start from like an apheresis, you activate the T cells, you introduce the CAR and the suicide gene, usually using a lentiviral vector, and then after this, you electroporate the cells and you knock out, for example, the TCR alpha gene, so you lose the TCR on the surface, so the cells are totally blind to non-self, so they will not induce any GVHD, and we had the proof of concept that our cells don't do any GVHD in patient. And then also you can alter other type of genes, such as, like, for example, CD52 to make it resistant to alemtuzumab. In our case, we made a pre-treatment with alemtuzumab, or you can, for example, alter other type of genes, such as deoxycytidine kinase to make the cells resistant to like a standard of care, such as like fludarabine, for example. Let the cell amplify, and then you have two key steps at the end, which is first filtration of the cells. So you have to get rid of everything you don't want. For example, T cells that still bear the TCR on the surface, and this is one of the key steps, the filtration, to get clean, non-TCR bearing T cells. And then you have like an automated fill and finish step where you can do vials at the quantity either for pediatric or adult use. And then once you do this, you freeze the cells, and you can ship them all over the world, 1 dose per patient, and usually can redose if necessary. And all this manufacturing has been proprietary developed by Cellectis. We believe that in the CAR T cell space, this is going to become one of the main key issues, being able to provide the technology to all the patients all around the world in a cost-effective manner. So everyone should be able to get the technology pretty easily in like a hospital that can treat them, and not only hospitals or like complicated procedure. And where the patient comes in, the patient can be immediately dosed. Not the patient has to wait for a month, maybe sometimes 2 months before the patient can be dosed. This is the way like this therapy we think should be conducted.
So as you've got, again, a unique perspective on things, and maybe you can help characterize some of these challenges from the Novartis lens, from a company that has the resources of Novartis. Are these daunting challenges? Are these basically, straightforward technical challenges? How does it compare? And then as you, again, look across the landscape, to what extent do you feel that, the industry as a whole is really where it needs to be on some of these considerations?
Sure. So I'm thinking about this sort of barrier to entry and building off some of the thoughts that colleagues have had here. So if I look back over 4 years, then what's happened over 4 years to make these treatments very, very tangible and tantalizingly close to reality? So first of all, building off the last panel data, clearly we were very, very excited by the initial clinical data, and for companies like Novartis to get excited off ends of 1, 2, and 3 is unusual. But we did, like everybody else. And then as we started to dig in, we now look back, we now have, together with our partners at the University of Pennsylvania, close to 200 patients worth of data now with CAR T therapies across different indications. So data certainly helps. And from those initial looks at the data, the real work starts. And this is where I'll differ a little bit with the previous panel and some of the thinking around the balance of clinical data versus manufacturing science data. So one of the things Novartis realized very early on was that the world of cell therapies, gene therapies, regenerative medicine, we have to balance the thinking in terms of a fully integrated understanding of clinical data, manufacturing science, clinical quality, attribute, and data, linking that back to research as well. And that was a big change management wake-up call for Novartis, because we traditionally didn't operate that way. Most of us have grown up in sequential drug development, 16, 15-year life cycles to get products to market. And here we're going potentially from proof of concept to commercial availability of a disruptive innovation within 7 years. Now, if somebody had said that to me 4 years ago, I would have said, not possible. But that gives you a sense of how quickly this field has moved. And some of the things that I reflect on why these things have happened, a couple of things. Firstly, capabilities, people. We were very, very fortunate that we had the vision and foresight of leaders in the company that decided to invest very early on in manufacturing science, which is why pretty much at the time when we announced our collaboration with Penn, very soon after we acquired a former Dendreon facility in Morris Plains. And that was a great win for us because of the people. We had legacy history of colleagues from Dendreon who'd done things incredibly well and also had learned lessons of how things, when they're not done so well, can lead to catastrophic business outcomes, which is unfortunately what happened with Dendreon, but let's not forget they were trailblazers, in my opinion, for having the courage of actually investing and developing great pedigree in science, particularly around manufacturing science. So integrating that clinical thinking with manufacturing science was a key thing for us. The third component, I think, was the scalability and availability of viral vector technology. Up to the recent past couple of years, it was very much a boutique cottage industry, very academically driven. But clearly now we're in a different era now where there are credible organizations, and we work with one of them very closely, Oxford Biomedica, where the ability to have scale-up but GMP quality viral vector was also a big shift and a big dynamic changed. And all of these coming together then in terms of further stronger clinical data, which was very exciting for us and others at ASH this past quarter, shows that we're in an era now where the next hurdles are now in front of us. And those hurdles, I mean, I can tell you simply the kind of things that keep me up at night where I spend a lot of my time focused on. Clearly scalability, as colleagues have said here. It will take a lot of investment and further efforts to scale up these therapies to treat patients around the world. Now, within Novartis, we are now successfully treating patients in the US, but also in Europe through our centralized manufacturing facility. That didn't happen overnight. That happened with a lot of investments in supply chain, logistics, understanding quality parameters, cryopreservation, apheresis, the whole enclosed ecosystem to be created. But we are not sitting comfortable with just version 1, right? We have to invest now in terms of process optimization, scale-up initiatives, aspects of automation. I don't personally believe that automation is the be-all and end-all. I think it's a finite component that will help. But actually improving transduction efficiencies of lentiviral vectors, looking at how you can reduce the needs on your source raw materials, how you bring down the cost of goods in a clinical manufacturing control setting without compromising quality, but then demonstrating those changes to regulators that they will not impact benefit risk is going to be the key. So on the one hand, we've been very successful in terms of tech transferring and developing what were academic processes to something that regulators are now comfortable with. And very much within Novartis, we are now on the next phase of evolution of how do we then make these fully available for patients around the world. So hopefully I gave you some thoughts on the kind of things that we're focused on and areas that we see as doable, but will require patience and tenacity and further investment over the next couple of years.
Very much so. I guess one of the things that strikes me is how fast the industry is flourishing. You know, what the panel members have talked about, it requires a lot of know-how. Where is all of this coming from? I mean, where are the people and the scientists coming from that are now able to populate such a vast effort in biotech? And, you know, Jeff, André, Gregg, you know, where are you hiring from that you're getting such talent?
Yeah, I think it's a combination of places, and it depends on what you're doing. So we actually happen to think automation will be an important aspect to success going forward. Maybe it doesn't have to be fully automated, but something that is automated closed system we think is critical. And we invested in that very early on and ended up collaborating with a company in Australia, of all places. And we're in North Carolina. And at the time, we had a partner in Japan who actually had been an early partner with Dendreon, who really was the one pushing us to invest in automation early. So We obviously would have chosen to work with a group that was closer to North Carolina if we could have, but it turned out that we believe the best engineers who are doing automated cell therapy are at this company Invitech in Australia. And then we hired internally engineers, some who had a lot of cell therapy experience, some who had very little, and then built our own team internally to work with the Invitech team. In North Carolina, we're lucky in that there's a lot of big pharma manufacturing expertise around that we can cherry pick from. So as we've grown dramatically over the last couple of years, we've been able to get people with not specific cell therapy experience to what our process is, but to some extent, it's all transferable in terms of that experience. So we haven't found it that difficult to find the people, but definitely you have to look at academia, you have to look at industry, you have to look at collaborators and kind of combine those.
Well, also that's a very important point. I think like talents in this space are pretty rare. It's a rare source. People that know how to manipulate lentiviral vectors in our space there are not that many. I'm personally like a retrovirologist by formation, so I've manipulated recombinant viruses back 20, 30 years ago. But there is not that many companies and that many labs that know how to make high-grade, high-quality lentiviral vectors, and this is a very rare source. So you want to rely on, there's not that many companies either. So you have to rely partly on companies and also partly on academic institutions that are pretty good to do this. For example, in the AAV space, there is a lot of very good academic resources for AAVs. You have some great companies also, but it's very rare. In gene editing, it's just a start. But one thing that you said that is extremely important is automation. And just remember one thing, back in the 80s, I was still a student in the lab, and I was doing manual sequencing, and people were asking me, André, when do you think human genome will be sequenced? I said, you're doing when you sequence 10 kbs, it was great success, you made an EMBO journal paper, and my answer was 2050, 2060 maybe. It was sequenced 10 years later, maybe 15 years later it was delivered. And the real revolution in biology, it's not that much. Computers, and it's probably computer sciences, but the real revolution in biology is automation. Automation cracked the human genome. Automation is solving high throughput screening. A lot of things are done in automation. Manufacturing is part of the automation. So heavy, and Cellectis is investing in this space, and the heavy investment in automation, fill and finish, as I said, electroporation, closed system, and all these things, and a constant evolution around this will crack the cost of good and accessibility of such type of therapies, and probably all the quality control over this. It's not going to be an easy task, but the path is there.
Yeah, Gregg, any additional thoughts?
Just a quick additional thought. You have to build a very different type of business platform in a autologous cellular immunotherapy company. The manufacturing, the clinical development, the R&D have to work incredibly closely together as you develop these cell therapies. Remember, the starting material for your cell therapy comes from the patient. So the beginning of your supply chain, if you will, is the patient and the physician that you're dealing with to procure the blood sample or the apheresis sample. So that means that manufacturing cannot exist in a silo as it typically does in many pharmaceutical companies. It has to be totally built in to the clinical team and to the R&D team as you move these products forward. And so it's a whole new structure, but it is absolutely required if we're going to move these cell therapies forward.
Yeah, it's a great point, which means that probably the most important group at Argos is the HR group, because getting all those people to work together is not the easiest thing to do.
So just to add, as well as the sort of, the excellent point just raised in terms of the integration, which I think I already mentioned, between the functions. I think now we're coming to the reality of the capabilities required to make these really commercially available. So the next phase of capability development in terms of people that we're looking at, for example, is how are we really going to have the capabilities in place with payers, reimbursement, market access, government affairs. Other areas that we look at now is sort of the service model. It's one of the reasons why Novartis took the bold step to look at this in a very focused way, in that this is just not the core business that many of us have grown up with. Life sciences, big pharma companies, we're not service companies. So actually appreciating what we can do really well and being humble about that, and what we don't know, was sort of the fusion of talent from external and internal, and obviously taking colleagues that had high agility, that had grown up in the world of genomics, biologics, that had transferable skills. And one of the big reasons why we acquired the Dendreon facility was exactly talent, people, because we knew we needed it. And now as we look forward a different phase of evolution, what are the capabilities we need moving forward? I mean, for example, we don't think in the world that we're moving to, we're going to need sales reps. We're not pushing a pill. We're not pushing a biologic infusion. We are dealing with centers of excellence, certainly in the autologous space, with bone marrow transplanters around the world. And their level of service needs are way different to anything any pharma has ever offered. So we are now focusing on the next level of capability build on how do you really make these therapies accessible, affordable, and available to as many of the appropriate patients. And we think autologous is a great beachhead to form. I'm admiring of colleagues in the allogenic space, because we also look at that as well, and how do we evolve to that in future years. But we believe that the beachhead now will be with autologous therapies, and how quickly can we get these available into as many patients as possible with new capabilities.
Right, and luckily for the three of us and the other biotech companies other than Novartis, I think the fact that this is a completely different business model for Big Pharma has scared most of Big Pharma away, at least for now. And because it is completely different from anything that Big Pharma's done before. And that's been the opportunity for companies like ours to step in and create that expertise, which didn't exist, doesn't exist. And even as you're thinking, as we're now waiting for our final phase 3 data, as I was hinted at, as you're thinking about commercialization, it's really a bottoms-up approach to commercialization, looking at your sites from your phase 3, and then going beyond that to those centers of excellence, because you only get one chance to launch, and you can't screw it up. And so, rather than looking at a top-down model, which is the Big Pharma approach to what's the total market size, and okay, how are we going to go? It's a bottoms-up approach to make sure you're working with centers that can support this, and then you go from there. So everything is different.
I'm not sure, unfortunately, we have much time for additional questions from the audience. Maybe I'll throw out one very quick question. Given the proliferation in the industry over the past 5 years, if you all had to guess, 5 years from now, as there's some potentially attrition, some consolidation, some new company formation, in terms of the number of companies and efforts out there, 5 years from now, will it be the same, more, or less than it is today? And Gregg, why don't we start with you and work our way.
We should make Oz go first.
Well, I would say that you'll always have, I think, a large group of development companies working with academic institutions, bringing these new cell therapies through the proof of concept stage. But I think in 5 years, you'll start to see consolidation around the cell therapy companies that can really manufacture. And I think what will happen is that these smaller development companies will take the product to a certain level, and then they'll have to turn it over to the manufacturers in this space.
Agreement, disagreement?
I'm not really good at predictions usually, on long-term predictions, but one thing I strongly believe in is the following thing, is I think we're at the prehistory of cell therapies in general, and also you have the conjunction of two things that are happening, which is gene editing on one side, and you'll probably see a lot of it this year. There is like new S1s that are filed for IPOs, and more technologies brought into the gene editing space, and gene editing combined with cell therapies gives tremendous amount of potential. So within 5 years, I think maybe there will be consolidation, but I think the number of opportunities to bring high-tech technologies and therapies to patients are infinite. So I think it will be a huge expansion. By the end of this decade, it will be huge, really.
Great. Oz, why don't we, just because we have a minute, Jeff, not to skip over you, but just in the interest of time.
5 years from now, 5 years, I think we'll have the reality for certainly a new pillar of medicine having been firmly established, certainly in the heme malignancy space. I see that with CAR T therapies, they will have come of age. I do see elements of consolidation happening as manufacturing science becomes, much more mature. As companies start to look at cost base very carefully, you will start to see, I think, some mergers occurring, some acquisitions occurring, consolidation occurring in the ecosystem, certainly with the companies that allow facilitation and enablement. I see a lot of excitement in the next 5 years around data. Data in the sense of the ability of companies to link data from benchtop manufacturing science quality to clinical and back reverse translating. I think that's going to spawn a whole new ecosystem around data integration that's going to allow even more opportunities for investors and for companies to flourish. So I'm personally very, very optimistic about where this landscape will be in 5 years, and I think the sector will continue to grow.
Well, this has been very helpful for me as someone who often perceives the innovation in this field to be a bit like a storm, not quite sure how to approach it or even where it quite came from. Maybe not the best questions, but terrific answers from everyone to really help understand where the field's come from and where it's going. So thank you all.